I'm Adam Dupuy. I'm an associate professor in the College of Medicine in the Department of Anatomy and Cell Biology, but I'm actually not a cell biologist. I'm a geneticist. I've worked my whole career in cancer genetics. Did my doctoral training at the University of Minnesota, and then postdoctoral fellowship at the National Cancer Institute before joining the faculty here. I've been working, so as my area of focus, I try to understand how mutation and genetic complexity contributes to cancer and all sort of the relevant questions we have about cancer. Why does it happen in the first place? Why does it progress and sometimes and not in others? Why does it metastasize in some cases and not others? When we go to treat cancer, why do some tumors respond and others don't? So these are all questions that can be addressed at a genetic level. And so my work really focuses on those questions and trying to understand them. Historically, understanding these questions and finding answers to them has helped craft more targeted therapies that have been much more effective in many areas of cancer. Ovarian cancer, unfortunately, as we'll discuss today, is one of the exceptions to this. We still don't have a clear understanding of the pathogenesis of this disease. So we're trying to gain entrance into this field with some of the pilot projects we've been working on over the past few years.
Yeah. You guys know Jane and I and my mom. My mom is sitting over here. Say hi, Nan.
Nan is here.
Yeah.
Nan.
Yeah.
But I want to say something on behalf of the team. We've been privileged and honored to be with Jacqueline from the get-go and Adam and the whole medical team. Obviously, this is a heartbeat passion for Phil and I, like our passion for life and adoption for Becca and just pro-life. This has hit our family hard. And I'm a person, Jane, but I'm a patient, Jane.
Yeah. Yeah.
And it's been pretty monumental. The past year, it's been exactly a year ago, I was undergoing chemo at this time.
Yeah.
And so I can't thank you all for taking time. Yeah. I have lost 9 friends that I have made on this journey in the past 10 years. I am the lone survivor.
Yeah. Yeah.
And it just takes my breath away.
Yeah. Yeah. So Jane and I would do anything. We'd jump out of an airplane to cure ovarian cancer. So part of my context here, and I sent Adam and Jacqueline my thoughts that I emailed you guys in terms of like, we want our trusted close friends around to help us really kind of figure out what energies and efforts this endeavor deserves. And, if someone told me if I raised $20 million to cure ovarian cancer, I'd friggin' raise $20 million to cure ovarian cancer. So I got a little bit, this isn't all about just raising money. It's about really kind of hoping to get, we've done enough base work on this thing and Adam's pivoted a little bit on different things. I think we're close to a point where we could drive this thing to a good concluding place that'll put us in a better position. So that's some context that I have, and then I'm going to introduce Jay here. And
okay, I'm Jay Olson, of course, friends of Phil and Jane. I even have enough Zoom knowledge to put a little heart up there while you were sharing there. So
he just retired from John Deere, and he's walked this journey along with us for a bunch of years. And
Phil asked, my role is, was a manager at the John Deere Technology Center, so my background's in farming, agriculture, and then on the engineering side, privy to a lot of projects that required, open collaboration with universities, mostly in the ag space. So I've been through a number of project reviews like this, and I sat in on the last one, Adam, that you did.
Yeah.
I congratulate you on making it through this last pilot, and Phil's summary is exciting, that you're close to into the validation part.
Yeah. Dave and Barb, you want to pipe in?
Sure. Good morning, everybody. My name's Dave Matisak, and this is my wife, Barb Matisak, and we are friends of the Allens and very interested in research and application. We recently have found out that we also have cancer in our family. My sister, Debbie, and our son, Andrew, his father-in-law, Mike. So we're extremely interested in learning and how we can apply this in our lives and apply it to others.
Yeah.
Barb, do you want to say something?
My background is as a nurse. So although it may be limited in the genetic area, but I do have the background that is helpful for understanding this process. So hopefully I can be of some benefit as well.
Yeah. And I think the other big context thing that I'm trying to do here, and Jay has a project in Uganda for farming that is basically sustainable charity too, that's actually worked. And I think all of us, all my colleagues, we all want to do something that's going to be impactful with our money and not just give it away to something. And 95% of the funding gets to the actual project, which is unlike a lot of the other ones. But anyhow, those are conversations that I've had amongst my friends and colleagues on this thing. So anyhow, why don't you Drew? Oh yeah, sorry, Drew, I forgot about you.
I think everyone knows me here, so I don't need to introduce.
Yeah. Yeah. So go ahead. Why don't you jump in, Adam? Adam, and Jacqueline, we had a conversation yesterday, and we framed out a rough plan, and hopefully we'll tune that up and so why don't you go ahead and take the lead on this thing, Adam?
Okay. Let's see. Can everyone see that?
Yeah.
Okay, great. So let me just start out as a preface here. So just so you understand the way these projects roll out. As I mentioned before, my lab has focused on a variety of cancer types, and usually to start to gain entrance into a new area within biomedical research, it can take years, 3 to 5 years of investigation before you have enough data to publish and compete for external funding. So each time I've entered into a new area, and we've done research on leukemia, liver cancer, melanoma, it takes a period of expansion. And so ovarian cancer is something we some years ago started work on in hopes that we could really move the needle for patients. There's a lot sort of critical need in this area. Let's see. And what I'm hoping to convince you of today is that there's a reason why clinical care for ovarian cancer has not moved at the pace it has for other types of cancer. And the reality is we still just do not have a fundamental understanding of the biology of ovarian cancer. So what I'm showing you here is sort of years of genetic analysis. This is analysis of hundreds and hundreds of tumors of these various different types: kidney cancer, colorectal cancer, brain cancer, leukemias, endometrial cancer, lung cancers. You're probably all familiar now with genome sequencing. This is a major new technology. We use it in my lab on a near daily basis. This is something that has really revolutionized cancer, our understanding of cancer for many forms of cancer, but not all. There are a couple types of cancer which fall out in what is called C class. And here the genomes are not mutated in the same way. So when you sequence them, it turns out it's less informative. And unfortunately, ovarian cancer and triple negative breast cancer are a couple of these. And because these cancers don't have informative genomics associated with them, it hasn't given us any insights into the biology of what drives these cancers to become life-threatening, like this information really has for these other types of cancer. Also, importantly, this is a hypothesis, mind you, but because the mutations that drive ovarian cancer are quite different from other types of cancer, I suspect this is also why immunotherapy has not been as effective in ovarian cancer as it has been in other types. But this is an ongoing area of investigation as well.
How many mutations are on a typical cancer cell for ovarian comparatively to others? Because the higher amount of mutations, typically the more treatable it is, correct?
Well, with immunotherapy, that's true, yes. So for a garden variety melanoma, cutaneous melanoma, there will be, 1 million plus mutations in a typical tumor. These are point mutations that actually change the proteins, and it's these mutant proteins that are presented to the immune system that elicit an immune response. And this is the real problem for ovarian cancer. The protein sequences themselves are not altered, generally speaking. In a couple of cases, that's true, but by and large, that's not the case. What changes is just the structure of the genome. So there's just more copies of some genes and fewer copies of others, but the gene sequences themselves are unaltered. And so the immune system likely has a harder time seeing that as abnormal. It also, because it's such a scrambling of the genome, it makes it difficult for cancer geneticists such as myself to really hone in on the specific mutations that are driving the process, because hundreds to thousands of genes are being impacted. And so it's difficult to distinguish which ones are causative and which ones are not. It's too many experiments would take far too long to sort through it one gene at a time. We basically have to test each gene in the genome for its role in ovarian cancer, and that is a challenge. So that's the sort of where we are starting. Now, other forms of cancer have also benefited from mouse modeling. And so let me just briefly discuss what that means. So, we are limited in what we can do in humans, of course, in terms of experiments. We can do observational studies, we can do genome sequencing. But in terms of genetic manipulation and asking basic questions about what mutations actually drive cancer, we have to turn to another organism, and often we turn to mice. The mouse is obviously quite different than us, that is true, but their genome is actually nearly the same size as ours. The genetic content is quite similar, and almost all of the genes in the mouse genome have a direct human counterpart. And so there are a lot of similarities between the human and mouse genome. The other advantages mice have, despite being small in size, they have almost all the tissue types and cell types that humans have, including ovaries, fallopian tubes, and the like for our discussion for today. And that is important. And so the technology that my lab works with to try engineer mouse models of cancer is this technology called Sleeping Beauty. So let me just walk you through. Can you all see my cursor here?
Yeah.
Adam has a published paper on this. Is just one published paper on it?
Oh, no. We've published dozens of papers on this technology over the years. Yeah. And we continue to have some coming out. Some we've done ourselves, others we've collaborated. But there have been, a dozen plus models of cancer in mice that have been developed using this technology that I developed as a grad student, actually, years and years ago. So what it is this little enzyme here, this little star guy here, SB. And what it will do when it gets into the nucleus of the cell is bind to bits of the DNA that are called transposons. Now, these are foreign. These are things we have to introduce. The mouse genome doesn't have these. But if they're present and this enzyme gets in there, it can bind to the ends of this thing and cut it out and make it hop, literally hop around in the genome of the cell. And what that can do is introduce mutations. And if you mobilize enough of these things, you can drive cancer. And so what this looks like, we used to do this by breeding. We combine mice, and then we could control where and when this process starts. all these transposons just start hopping around in the genome. This is a little cartoon of a genome here. And this happens differently in all the cells of the animal. But if we do a good job of this, get the efficiency high enough, we can get a cell that gets the right combination of mutations to transform it or turn it into a cancer, and it will grow out. And then all we have to do is go in and identify where all these transposons are at in the tumor genome and identify all the genes that are responsible for that particular tumor. It's a way for us to accelerate the discovery and identify the important genes, and then focus in on the biology of those alterations and their role in cancer. And this has been helpful in a number of the models that have been developed over time. And so years ago, it was our original goal to develop ovarian cancer models using this technology. And we tried. We struggled with this for years. The goal was, if we could identify genes that drive ovarian cancer in the mouse, we could compare that to the databases of human ovarian cancers that have been generated. And anything that's common between the two species will be the genes we would focus on. These will probably be the genes that are important. If it's an alteration in a mouse ovarian tumor and it's an alteration in human ovarian tumor, chances are that's not a coincidence. It's probably because it's important to the biology of ovarian cancer. And so then we would focus on those in terms of therapy and further experimentation to understand how these things really contribute to the biology of ovarian cancer, which is where we really need to push. And so we tried for years to do this. We tried 3 different major efforts to get this to happen. And so with mice, you can do this in a couple of ways. You can introduce mutations into specific target cells, as I just described, and this is what we tried. But there are other options. You can expose mice to carcinogens to induce cancer. This works well for some types of cancer, but not for ovarian. This is really not a good option for ovarian cancer. But then you can also
talcum powder.
Maybe, yeah. So that does increase risk of ovarian cancer, and it is something we could do. That's a long-term experiment, though. It's pretty chronic exposure in humans that it's associated with increased risk. And then finally, the final option, which is eventually where we landed, is we can introduce tumor cells directly into mice. And so more recently, and I'll show you data today from this effort, we have engineered human ovarian cancer cell lines and are now making pretty good, actually fairly rapid progress with this approach. But first, let me talk to you about our initial efforts. So what you're seeing here on the left is sort of a cartoon of the female reproductive tract for humans. You see the ovaries, the uterus, cervix. And on the right is the system for mouse. So here you see the uterus in mice. The uterus is more of a Y or V-shaped organ. The ovaries sit right at the top in these very, very small fallopian tubes. Now, a mouse ovary is about 1 millimeter in size, compared to about 3.5 centimeters in humans. So this is a small, very small target. We tried inducing mutations in these sites by direct viral injection. This required surgeons coming into the lab, seeing if they could get it done. It was not reproducible. There have been a couple publications on this that people have done it, but most labs publish one effort and then struggle to maintain that model. It's quite technically challenging. So we abandoned that approach and then went to 2 different methods to try and induce mutations in these sites in mice. And at the end of the day, this is after years of struggle and hundreds of animals, unfortunately. What we determined was that the genetic tools are just not out there to specifically target these cells. So these cells are epithelial in nature, and there are no tools that can target them specifically without also inducing mutations in other epithelial tissues throughout the body. And so what we found is what others have also found. In the process of trying to induce ovarian cancer, what you end up doing is inducing all kinds of other cancers in mice that they get much more quickly and much more efficiently. And so we had to contend with this tremendous background of a whole host of different types of cancers. And so so we kind of decided to go back to the drawing board and think about what we could do to move forward after this process failed. And so we've rolled to this xenograft model, or this model of engineering human ovarian cancer cells and putting them into mice. And so we've made a few alterations to these cell lines to facilitate our efforts. So one thing that we can do, we want to be able to track disease progression in vivo without having to euthanize the animals along the way. So we want to do, much like in human patients, just imaging throughout and see the tumors progress. And so we can do that by genetic engineering of the ovarian cancer cells here. There's this sort of neat trick. The gene that makes fireflies light up, it's called luciferase. That gene's been cloned, and you can express it in cancer cells. And if you put those cancer cells into mice and inject them with the substrate for this enzyme luciferase and put them into this closed dark chamber, you can actually see the luciferase signal coming from the tumor cells in the animal directly. And so we can visualize the tumor cells as they grow over time. And so we've done this with 3 different ovarian cancer cell lines. One of them we knew would grow out aggressively, and that's this one called ES2. And so when we injected these cells into the intraperitoneal cavity of the mice to mimic sort of intraperitoneal disease, ovarian cancer, you can see the signal here on day 1, just after injection. Day 5, the tumor is already growing. As the cells get more abundant, the signal shifts to red. And you can see by day 15, the animals have already developed sort of ascites and sort of the features of aggressive disease. And the signal is quite strong. And so this helped us sort of establish all our techniques. Now what we've done is we've rolled to 2 ovarian cancer cell lines. The first one is OVCAR2. You can see the animals here. Here, the tumor cells are there. They're very weak and dim, not very abundant. And they don't really grow that well in the mice. And this is good, and for reasons I'll get to in a bit. And we saw this as well with the second model. And so we have 2 models now that will sort of persist in the animal, but will not progress. So you could view these as sort of early stage disease. And so if you quantify this, you can see on the left is the aggressive model. You have to look at the scale. I'll show you a combined graph in just a second. The cells are still there for the less aggressive models. Hopefully I can have convinced you of that. But if you compress them onto a single chart, here's the aggressive model, and here are the other 2. They're present, but not really growing. So now, what do we want to do with this? What we can do now is we've developed over the past few years, we've developed a new version of the Sleeping Beauty system. We don't need to engineer the mice directly anymore. We can engineer the tumor cells specifically. And actually, this has allowed things to accelerate quite nicely. So now what we've done is we've engineered these cells not only to express the firefly enzyme, but they also express that enzyme, that transposase enzyme that I talked about earlier. And so now we can just introduce the mutagenic transposons into these cells, inject them into the mice, and then image weekly. And what we hope to see is that these tumors will grow out like this. And what this will show us is that the transposon-induced mutations have driven progression of the disease. And if this works out, then we can go in and quickly identify all of the mutations associated with disease progression. Now, that's just the starting point. Once we do the genetic analysis, we've also made some additional technological developments over the last couple of years, including the ability to analyze these cells at a single cell level. So being able to do single cell genomic profiling on these tumors is giving us much greater resolution to see what's going on in the tumor as it develops. And so this is a combination of a microfluidics platform with some fancy reagents that end up getting encapsulated in little droplets so that we can do single cell amplifications and barcoding here. And then we sequence these things in mass. And then with some sort of intensive bioinformatics that this is my contribution to this project. I also do bioinformatics, computer programming. And so we can actually piece together, starting with the initiating cell that's introduced, we can kind of see how that cell progresses and the mutations it acquires and how that affects tumor growth and response. And so we can piece together sort of the genetic evolution of this tumor in the animal. And so we can really get fine detail about the mutations that are associated with disease progression. And so we have all these systems now in place. They're up and running, and we are ready to go to do this pilot experiment. We're going to start in at least a small scale next week with this, but we would need to expand dramatically to get sort of a definitive result. And so that will take a significant financial investment. This is what I project we would need for a 12-month period to do this experiment in total. It's not inexpensive, and as you'll see, personnel is the most expensive part. This is something that I think not a lot of people understand, that even though I am a faculty member in the College of Medicine, everyone in my lab are technically employees of the College of Medicine. The College of Medicine actually provides us nothing in terms of financial support. I have to pay everyone separately. So I have to find ways to bring in money to pay my own people, even though they're technically employees of the College of Medicine. So it's a constant sort of hustle to find resources to maintain the staffing of my lab, in fact. And so where we've gotten thus far with the ovarian cancer project is by me convincing people to do these additional experiments on the side, in addition to the experiments that we're funded to do, that are basically covering their salary. So we've moved at the pace we've moved with basically the personnel I have in place. To expand and move quickly, we would need to bring in new people, and that is a huge cost. And so we can continue to move at the pace we have, but it would be nice to really focus, have someone on these projects full time. That is my goal for the coming year.
How many? What's your hey, Adam? What's your personnel? And then it's one thing to get people in place to do this short-term stuff, but to keep them in place takes additional funding too, and that's the other.
Yes.
Piece
yeah, to keep people long-term, that's the real struggle. Of course, all of these folks are highly trained, and they're looking for, not surprisingly, some stability in their work.
Yeah.
And so we really need to get external grant support to get this to be sort of a stable, ongoing project. And so with these pilot projects, we need in the next 12 months to get a grant funded. We came close last year, actually. I had a grant on ovarian cancer with the Sleeping Beauty approach that was selected for funding through a Department of Defense mechanism. We were selected for funding as an alternate. And unfortunately, the DoD ran out of money. That was during the pandemic year. Yeah. And we didn't end up getting those funds, but we came close. So that was encouraging.
So, hey, Adam, talk a little bit about the grant funding. Okay, so you're looking to have like 30 mice that you're going to like pilot this thing, and then that gets you a pretty good data set, and then that gives you enough like ability to write a more extensive grant. And then what size of those grants be, and what's the, how long would that take you? I mean, what would be your, I know you can't say definitively, but I'm just trying to get my arms around, like, if we invest in the 183,000 now, what's our.
Yeah, so a major NCI grant would bring in somewhere around $250,000 a year for 5 years. And so for us, in science, we have a sort of competitive system, just like everything else. And so we compete with each other for funding. And so even when we get a grant funded, it's for a specified period of time, and then you have to go in and compete again to get additional funding. So it's a 5-year cycle, generally speaking, and we're looking at around $250,000 a year.
Yeah. And what would that $250,000 a year get you in terms of like starting the project now with by hook or by crook, with the money raised, and then the 5-year, you got $250,000 a year.
Yeah.
How much would that accelerate your?
Yeah. So with that budget, I could bring in probably 2 full-time people to work, only on this project. And so the pace at which we could move and the expansion and the focus would be tremendous. So once we do the pilot and identify some candidate drivers of ovarian cancer progression, we can also go to the tumor bank here and do experiments to evaluate these candidates in ovarian cancer materials that we have banked through our hospital and see. This is the process we go by. We identify these things in models. We go back to the patient samples and see, did we identify something that seems real? Are these things also altered in human tumors? If the answer is yes, then we really focus on trying to understand how we can disrupt this process. We found something that drives the disease forward. How can we disrupt it? Is there a drug that would inhibit this process? If so, can we do a clinical trial? That would be another effort that we would have to go through with the gyn-onc colleagues we have here, including Dave Bender, Mike Goodhart, write a clinical trial and actually test. That would be our hope. For me personally, my preference, if we could do it, I can give you the example of high dose ascorbate. This is something I've talked to Dr. Bender about trying in ovarian cancer models. There's some evidence that oxidative stress is a major issue in ovarian cancer, and maybe it could be exploited with high dose ascorbate. It's a very safe, relatively inexpensive therapy. That's what I would prefer.
Vitamin C.
But it's hard to get that. So we can't get a corporate sponsor for that clinical trial because no company can get intellectual property on vitamin C. So we have to raise money and get trials funded through the NCI to do those experiments. For the patient, that would be better. It's a cheaper therapy. And if it's as effective as something that a company can generate, I think that's best. Something a company would generate is going to be patentable and the cost is going to be extremely high.
Yeah.
That's just the system we have.
Yeah.
So if we could find a way, if we find genetic drivers of disease progression, and I could block those processes with drugs I could get off the shelf at Walgreens, that's what we're going to do. Because that's our goal, is to find effective treatments that can get to as many people as possible. If that's not possible, then we're going to go to FDA-approved drugs, because that can accelerate things. If there's something already available that's FDA-approved, then we'll go there. If there's nothing there, then we have to talk to collaborators in the College of Pharmacy to develop a drug. It's a longer-term process. We'll do that if we have to. We try to find something that's available because that's faster.
Hey, Adam, how many laboratories across the country are doing mouse modeling of ovarian cancer?
Hardly any. Hardly any.
Mm-hmm.
There are only a handful of models that have been described, and most labs will publish one paper, but you never see anything more from them. And what we always wondered why that was until we started trying to model ovarian cancer ourselves and figured out that it's highly inefficient and not very easy to do. So it's a real problem, and it's part of the reason I think part of the reason that treatment has not progressed like it has in other cancer types is because of the lack of good models of this disease. Yeah. We still don't have a good firm understanding of what the biological processes are that drive ovarian cancer specifically. And until we can get that, until we can answer that, I think we won't be able to really make rapid progress.
Has anyone used other animals like pigs at all?
No pig models I'm aware of. There are some very old school models that have used rabbits, oddly, but those haven't really translated into the modern era. I don't know of anyone actually using those now. There are other types of models called PDXs, which are patient-derived xenografts. So instead of, we have cell lines, these models would be taking tumor material straight from a patient and putting it into a mouse and trying to propagate it in an animal without putting it into cell culture. We could try some of those models as well. But in terms of a spontaneous model of ovarian cancer generated in mice, there are not very many that have been described.
Okay.
Yeah.
Are there other cancers besides ovarian that currently would use the underlying sleeping beauty technology, as well as the other barcoding technology that you have developed?
The single cell stuff?
Yeah.
Yeah, so we're the only ones doing single cell. We developed that technology here, so that's uniquely ours. We're working with a couple of companies to develop this approach. We're hoping to publish it late in 2021 or early 2022.
Okay.
So I think it'll be, with the data we've already generated, it's going to be provide a lot of insights that we were blind to before, just because of the way we were forced to do the genetic analysis. So it's been quite transformative for us. I think it'll be a huge advantage for us as we move forward.
So, hey, Adam, is there any like hard stops, like 6 months, if you got like a bucket of money, say in December, and you could start this thing? Are there any hard stops out 6 months that gives us kind of a place to like recalibrate and see if we're on track? And
yeah, well, I mean, for us, and thinking about our path forward with ovarian cancer, as long as we can continue the work, for me, there are no hard stops. If we hit a technical hurdle, we'll just always pivot, which is what we've done to this point.
Yeah.
I don't envision we'll hit too many hurdles. We have the 2 cell lines. There are a dozen or more other ovarian cancer cell lines that we could try. So we can continue to engineer and try one until we get something online. That would be our first effort.
Okay.
It may slow us down, but it won't stop the work.
Okay. Okay.
I'll jump in for a second, just with my like kind of fundraiser hat on, to kind of just like talk about the process in a sense too. Hi, Ralph. This is Jacqueline. I don't know if you can sing. So my office, and I know, those of you all here that have philanthropically given and know that a gift of philanthropy, then you would designate, and as Phil and Jane have said, the funding that would go, for example, toward ovarian cancer research, to go to the lab, for personnel, for all those things that Adam has indicated, that is something that goes to a said fund that then you say, this is my gift. This is a gift of philanthropy that we believe we, feel really good about the work that's going on. And from there, the process, whether that's Adam collaborating then with Goodhart and Bender and kind of getting that baseline of like, hey guys, these are the next steps. This is what we're going to do. If there's additional funding coming in, that eventually, as with a back end, would have some time to get to them to start. Then for Adam to kind of pull in, who are these personnel? When can they start? When can I get them on board? So, timing takes a little bit as well to get things in place and get things rolling. And from there, part of, my work with Adam and my work with all of you is to continue to keep people updated in what we're doing. Or if Adam's like, hey, this is super cool, we've just kind of hit this really cool mark right now, this is definitely worth sharing. Yeah. So to give him and his team that, ability and that freedom to do what they do best. And then, for us as donors to be part of that experience, knowing that that funding is there with him, and will only be used for what it's designated for. And in time in working with, Adam and the world of science and researchers, I mean, there's always hiccups, there's always roadblocks. Science takes a long time. It's like, as he said, it's like you're looking at a mouse every week. And it might just be looking at a mouse and seeing what you see. And 3 months later, it's like, oh, cool. So I think it's kind of setting that expectation of like, we know it's going to also take time. and the resources with that. And so I think the most rewarding thing, at least in working with donors from my end, and is seeing the results over time, and for people feeling really good about what they're doing and that it is really making an impact on people like Adam, to be able to find other people to put in his wheelhouse that he works with to actually do this work. Because, as he said, one of the things with so it's the hospital and the College of Medicine, they're essentially combined as like you can walk together through them, but the College of Medicine then, where all the research is taking place in the labs, I mean, there's people in those labs that can work on, for example, different diseases within cancer, but they're going to work where the funding's available because that's their job. That's what they have to do. And so if we can find a way to continue to say, this is really important, and we can pull people in for ovarian cancer, just ovarian cancer, that's a gap that needs to be filled, because there's a sense of urgency, and to be able to allow him to pull those smart brains in, and Adam said too, and Phil and Jay know this, that like we recruit people from all over the country to come here because their brains are so awesome and amazing and can do this work, but we have to have money to keep them here. Yeah. And we might be able to do a project for a year once it gets going, but then once that's done, if we can't keep them here, we can't keep on with that. And that's, and that's everywhere throughout the country at every institution like this. So,
Hey, Adam, when is the next grant cycle? I mean, if you were to get a bunch of this work done, when would the next grant cycle can be? Because we get national science. We've gotten two National Science Foundation grants for our Perceive entity, and there's sort of a cycle on that. But what's the timing on the grant stuff?
Yeah, NIH, NCI grants, the general deadlines for the major funding mechanism is October, February, and June.
Okay.
Yeah, so I just am submitting a grant today, in fact, actually, for the October deadline.
Yeah. So would you look at submitting a grant on this maybe in June if you had enough stuff?
That would be the goal, yeah. If we can move and we hit, we could probably put something together for the June deadline.
So you would be able to create enough data and enough backup science to be able to get a subsidized enough grant?
Yeah, so one advantage of this new approach we're taking is it is much faster, much faster. So before we had to generate the animals by breeding, which is a slow and inefficient process. It took us years to generate the mice, age them, monitor them, see what sort of diseases they were getting. Here, we know they're going to get ovarian cancer, and we can generate them synchronously all at the same time.
Yeah.
And we get it done. And if SB does the job we expect it to do, it'll drive disease progression rapidly, and we can acquire all the materials in weeks. And then the sequencing can be done within weeks as well. And now we have the pipelines in place, and our.
Did you use a company on the West Coast to do that? Did you tell me, or?
Oh yeah, we're working with a company called Mission Bio to do the single cell stuff. Yeah, we have their device. They gave us a device. We've got it sitting in the lab. So we've been working with them to adapt, to generate a protocol to use on their platform. Once we have all the material, we can move very quickly and do the analysis. That only takes a couple of weeks.
Oh, okay.
Yeah, we can move quickly. It's just so we can move quickly if the financial resources are there to facilitate it. That's really what it comes down to. Well, the facilities here have the capacity to do the sequencing. They can do it. They do runs every week. We could fill the machine every week.
How long does that take to do the sequencing out of those machines?
Over the weekend, we can generate hundreds of millions of reads. It's really quite fast.
Yeah.
We could submit samples on a weekly basis and generate terabytes of data on an ongoing basis. And with our high capacity computing facility here, we can analyze it quite rapidly.
Back to you, Jacqueline.
I'll unmute again. Back to me.
Yeah.
Hi. So,
Want me for interrupting?
Yes, I was not a happy.
no, it's
wife.
fine. I think I mean, if anyone else has any like questions, comments, thoughts, for Adam, for myself, I know that this is a lot, and you know, and I think in the big picture of this, you know, number one, I mean, we truly, genuinely appreciate your interest in this, and any support makes a difference. And I mean, it takes a lot of support to get, you know, what we would envision like way down the road. And the hard part too, and what I've, you know, learned over the years too, like with medicine and with research, with cancer specifically, there's no end. There's never like an end. There's never a final, there's never a product. There's never that thing at the end. There's always the journey. And you can't even start the journey without, I mean, really without private support. And, you know, as Adam tells me a lot, like in talking to him this month on and off, he's just like the amount of, you know, applying for grants, the amount of time it takes to apply for grants, it takes you away from even being able to do the littlest things. And so having funding for pilot projects, for getting something even to the start remotely of a phase 1 to continue that, I mean, eventually to have notoriety, to have that national recognition and have that larger grant funding in the end is, you know, it's so important, but it takes a really, really long time and a lot of money to get there.
I should also mention, of all the institutes within the National Institutes of Health, NCI has one of the lowest, worst pay lines for grants. They fund less than 10% of the grants that are submitted every cycle.
Yeah.
So it has been throughout my entire career as a faculty member, a constant battle for NCI funding. My colleagues in other areas of disease research, you know, have pay lines that are, you know, upper teens approaching 20%, something a little more manageable. So it's a constant struggle in the cancer realm for funding.
How do they determine? I mean, is it segmented by cancer types that they allocate, or is it just best proposals?
Yeah, occasionally there'll be some congressionally mandated funding mechanism on one type of cancer, but generally speaking, that's rare. Usually you get all comers for all tumor types, all areas of cancer biology, all sort of competing simultaneously. Yeah.
Okay.
Now, there are private fundraising groups, as I'm sure you're all aware. Ovarian Cancer has a number of them that raise funds specifically to fund grants for ovarian cancer research. You know, they can be helpful in getting grants for pilot projects. They will fund grants for pilot projects. The National Cancer Institute is notoriously bad at providing funding for pilot projects. If you want a grant from NCI, you have to go at them with a lot of data. So, you know, it's just the way it is right now.
Okay, cool.
And I also want to mention, and Phil and Jane know this, like, I'd be very happy to also have like side off conversations with everybody too, just to kind of drill in and like process wise, what that looks like, what, you know, people in general who might be interested, donors to, you know, various areas within the hospital or wherever within the university, what that looks like and what that process is. So I would be, if I didn't mention that, at least I'm very serious about that, regardless of the outcome of that. Like, I'd be very happy to talk to everybody about that too, one-off, just, and what that means, what that looks like. So you know that.
Hey, Jay, did you have a comment or? Oh yeah. You're muted. You're muted.
There we go. course, I'm, an outsider. When I only was in the last meeting, of course, I get to talk to Jane and Phil. But so it looks like the next phase is if you can find a correlation of mutations to the genotype that identifies an opportunity for what's the right word?
Engineering something that's going to block it.
Yeah,
right?
so that then you can get to the next phase, which is the immunology part of it, which chemicals would slow that growth. So this next phase is really developing a standard test method that which you've been very clear on from the beginning, but you're at that point now based on what you've done so far that there is an opportunity there that looks promising. And again, it's the next stage gate, which is having a standard test method, right?
Yeah. And in terms of drug development and testing in ovarian cancer, a lot of the small sort of steps forward that have been made, there are drugs that have been approved for other cancers that gyne oncologists just tried in ovarian cancer patients, like Avastin or these other proteasome inhibitors.
We're on it.
Yeah. So those were rationally designed to treat other forms of cancer. And once they got FDA approved, that allows physicians to try them in off-label use and just see how they perform. And that's essentially how this progress has been made in ovarian cancer. And that's because we don't understand the biology of ovarian cancer. So it's just sort of haphazard. We'll test these things and see if they work.
That's the shotgun approach, right?
Yes. Yeah, exactly.
Hey, Adam, I think we had this conversation last time, but don't you have some other examples of these really strange things that block stuff? And wasn't one of them you talked about in melanoma?
Yes, MEK inhibitors.
Yeah.
Yes. We have, so I didn't have slides for it today, but we're also testing MEK inhibitors for their ability to treat ovarian cancer.
Right.
There's actually a trial that's not yet published, but a drug called trametinib, which is an inhibitor of this enzyme called MEK. It's approved, again, in melanoma, but it has shown efficacy. It outperformed the standard of care for women with a particular form of ovarian cancer called low-grade serous ovarian cancer. And so that's quite promising. So we're looking to identify resistance mechanisms for that drug.
Would after a year, if everything, if there's some acceptable correlation, or whatever, that shows that you're ready to go to the next phase, that testing of chemicals or drugs, whatever, would be on the next phase would be on mice.
Yes.
Then if that looked promising, then you would go to clinical trials, right?
Yes.
Okay.
Well, if we chose to do that, if we think the data are compelling enough and it's an FDA-approved drug where the safety is already worked out, we could actually do those things simultaneously.
That's good.
Yeah. So if it's a novel compound that's never been in humans before, animal testing is the first step along the way. But if it's something like bevacizumab or Avastin, something that has been in patients and the safety's been worked out, well, you can go right to clinical trial, actually.
Yeah. Yeah. Hey, the other thing is, Adam, is Ralph here, his sister is Margie Zimmerman or Margie Agnew, and Adam's the Tips for Tad connection there. So
for melanoma.
Yeah, for melanoma.
Yeah.
So I Ralph, I knew that example. I didn't bring that up just to do that but I knew that there's a couple of really good examples of stuff that's actually worked in similar cases on other cancers. So,
yeah. And we, I have to say, another hat I wear as the director of our cancer biology graduate program. This is a new program I developed a few years ago, training students specifically in cancer biology. And I decided through, my interactions with patients and their families, I actually require my students to shadow physicians in their second year. We really feel strongly in motivating people to, science is tough and it's at times a challenge, and it's helpful when you have these interactions to remember why it is we do what we're doing.
Yeah. Yeah.
Hey guys, I have to drop. Thanks so much. I appreciate it.
Yep.
Thanks.
Bye,
for your
honey.
time.
Love ya.
Great to see you,
Yeah.
Drew.
Bye.
Yeah,
Bye.
I'll reach out to you guys, and then I'll send a follow-up email of the link that we've set up for the donations, and then whatever, I'll follow up with some other information. But my other colleague, Joe, I'll send him this recording, and he's very interested, and we can hopefully figure out a way to get this thing done, so so anyhow, so much. Thank you so much for your time, Adam. It's just been a pleasure to know you over the years, and
thank you for working your efforts tirelessly, diligently,
yeah.
a man of perseverance. Thank you.
All us business guys recognize the value of a guy that wakes up every morning and figures out how to sell circuit breakers, right, Dave?
Yeah.
And how to
Yeah,
do
we
it
do.
and how to get it done.
Right, and how to solve the ovarian cancer puzzle and all that kind of stuff. So,
Yeah, I think about this stuff constantly. I come up with my best ideas mowing the lawn most often.
yeah.
Well, Adam and Jacqueline, Barb and I enjoyed meeting you, and we gathered a lot of information, and thank you very much for presenting it to us.
Yeah, thanks.
I'm going to figure out a way to get this thing done for you, Adam. So,
All
We're
right.
working on it piece by piece, Adam.
yeah. And
Hey, that's how we do it.
You'll allocate my mom's donation to this one too? Okay.
Yes.
Well, that's exciting.
Yeah.
I was looking over the list and I'm like, whoa, that's gotten. So
Thank you guys for everything you do. We
Right.
appreciate it very much.
Love to
Feel free,
all.
you guys, too, to reach
Bye.
out if you have questions
Bye.
and I can help.
Bye.
bye,
Excellent.
you.
guys. Bye.
Bye, everybody.
Thank you so much.
Thank you.